All right, well, thanks everyone for coming. It's good to see a lot of faces I know and a few new faces, and hopefully I'll get to know you afterwards and find out why you're interested in Raman microspectroscopy. So this is a MONT seminar. It's being recorded. So if you ask questions later on or in between, please let me repeat the question so that other people can hear it that might be logging in remotely. And then I will hopefully answer it properly. So you've all had a chance to look at the title through the announcements. This title is the result of mostly the users of this instrument who come from various disciplines. What I really want to do today is to talk to you about Raman spectroscopy and Raman microspectroscopy. Some of the cool things that we've done. And when I say we, it's a royal we. It's really a lot of you who are sitting out there right now and are probably remotely logged in. Okay, so the new Raman, as I called it until recently, is in the RASCL, which is a laboratory in Barnard Hall 331, and RASCL stands for Raman and Single Cell Analysis Laboratory. If you have the chance to walk in there, you'll see this instrument, which is a LabRAM HR evolution from Horiba. I'll show you later on. There's another one now, which I'll talk about details later on. It's a microscope, and when I got it originally, I was very surprised. There are no oculars on there. You cannot look through it with your own eyes. It's all via camera. So originally, they were afraid I was going to send it back, but I guess that's the new world. You don't need oculars anymore. I will refer to the scope as the Raman Scope, the Raman Confocal, the Raman Spectromicroscope. So they're all exchangeable, and you'll hear various people use these terms in different contexts. This was not a purchase that I could have made alone. There's a number of people that have been involved, and institutions, including MONT, the Energy Research Institute, the CBE, as well as Connie Chang, Jim Wilking, Al Cunningham, Colin Shaw, and the ICAL. And the ICAL, as you will hear throughout this talk, is pretty much essential, because just like any other instrument, the Raman is not going to give you all the answers. It's just one tool in the toolbox, and hopefully I'll be able to explain to you what this tool can do. Not necessarily demonstrate everything, but at least explain to you what this tool might be able to do for you. Okay, so Raman spectroscopy is based on inelastic scattering of mostly monochromatic laser light. When this happens, the wavelength changes, and you basically record the spectrum of the emitting light. And this happens because vibrations occur in the bonds, and then the molecules go back into a non-excited or only partially excited state. So here's a carbonate ion, and if you shine laser light on it, you have a lot of elastic scattering or Rayleigh scattering, and about 1 in 100 million scattering events is inelastic, and that causes this Stokes scattering that we basically record the Raman scattering. So what we then do is we take in order to get these spectra, we take a laser source, and this doesn't have to go through an objective into a microscope. There are also Raman spectrometers, handheld, that you can hold and you get a spectrum back. But if you have a Raman microscope, you take a laser source, you go through an objective onto your sample, you excite the sample, and then you collect the light that's coming back. You collect a spectrum through a spectrograph, and then you have a computer to actually analyze the spectrum. On the left here, here's two. Again, this is the scope that we have. This is an entry-level instrument. What's important here, especially if you ever use this, is to keep ambient light out, because that can really affect your spectra. We do this by actually turning off the lights and the room is darkened. And we need sometimes open access to the microscope. Okay, so I talked about lasers. So this is a graph that shows you if you have, there's 3 different laser sources here, and we basically collected the Raman spectra. What I'm plotting here is intensity over wavelength. This is not how we usually analyze spectra, but I want to start that way. The way we usually display spectra is through the wave number, which is in 1/centimeter. And basically what you do is you divide 1 over the initial wavelength, minus 1 over the wavelength that you're reading out, and you calculate a wave number. And I'll come back to that in a minute. But what I want to show here is, so if you have a laser at 473 nanometers, you get a spectrum, a Raman spectrum, or I should say you get a spectrum first. And if there's fluorescence, that fluorescence might totally extinguish or overlap your Raman spectrum, so you might not see all these peaks. In this case, you can, but what you see here is you have a response of a certain wavelength range. If you take a higher wavelength, a very similar response is stretched out over a longer wavelength range. And again, once we plot it in inverse, the wave number is going to be the same for all these peaks, especially this big one here. Oh, I'm sorry, the big one is not here, over on this side. But so the lasers allow you to stretch out the spectrum, but only to a limit, because back here, when you use 785, you stretch it out so far that your detector actually can't get this peak anymore. You're beyond the spectral range of your detector. So you got to be aware of that. And the other part that I already touched on, if you have fluorescence, for instance, we work with GFP a lot, green fluorescent protein, which we excited about 488, we would have a huge peak here that would basically not allow us to see any of the Raman peaks. So that's why multiple lasers can be important. And we often test them before we actually run analysis. So, but what you do, if you have a good spectrum, you take the spectrum and you either compare it to an existing spectrum, and we have access to what's called the know-it-all Raman spectral database, and everybody at MSU has it or can get access to it. Just let me know, and I'll let you know how to get access to it. Sometimes you run standards, and sometimes you compare your spectra to controls, and I'll touch on all those. To keep in mind for all of you probably, if you don't know it yet, it's non-destructive. There's very little sample preparation that you need to do. You take your sample, you put it under the scope. For SEM and other high analytical techniques, you usually need to dry it, put it into a vacuum, freeze it. For Raman, in principle, there's no sample preparation. And so it's non-destructive. You can do that, and then you can go to the SEM, for instance. So this is kind of the background. Hopefully I've got everybody on the same page. Now I want to give a shout out to a bunch of you who've actually done a lot of work. So I have a research group that works, among other things, on infectious kidney stones, struvite stones. What happens there is, if you have a urinary tract infection, the organism hydrolyzes urea, produces ammonium, and in the presence of magnesium and ammonium and phosphate that's in the urine, struvite, magnesium ammonium phosphate, can precipitate. For those who are not familiar with it, these are called staghorn stones. They fill out your whole kidney. Sounds painful, right? And this is how they look. Now, what we're trying to do is we're trying to understand what the role is of organics and biofilms there, and how mineral formation, meaning stone formation, can be potentially prevented. So I've had the chance to work with a lot of really smart and hardworking folks, Trace Hobbs, an undergrad, Logan Schultz, now a professor here, and Erica Espinosa-Ortiz, a research professor here. And we basically built a model system of the urinary tract using reactors, tubing, a capillary that we can look at under the microscope, and then a bladder which can be emptied. And up here, there's some sponges that we put in as tissue equivalents. So we can look at this, and if we look at it under the confocal scanning laser microscope, we can get these beautiful images. Green is mostly cells, and then you can get reflection images to see hard surfaces, the mineral. If we put this under the SEM, the scanning electron microscope, we see these beautiful struvite, and I call them struvite-like crystals because that's how struvite often looks like. But we have to destroy the sample. We have to stain it for confocal microscopy, or we have to put it under a vacuum for scanning electron microscopy. What we can also do, and I pointed out this capillary, we can run the system through this capillary and then look at it under the scope. And we did so, and what you can see here is minerals precipitating in a capillary. So we took those and then we tested out two lasers in this case, 532 and 638, and decided, which one gives us a better spectrum. And that's initially somewhat subjective. What you're looking at is which wavelength range you are, or which wave number range you're interested in. So the Raman shift, the wave number, is really what we are looking at. And in this case, we were mostly interested in this area here. So here the baseline was much straighter, so we decided to use the 532 laser. So what we then did is we looked at different parts of the capillary. The glass, the water, close to the glass, some of the liquid inside the glass, and then closer and closer to the crystal, we started seeing this peak here, as well as a few other peaks. But this peak was of specific interest to us because the 945 is an indicator of a symmetric stretch in phosphate. So this indicates that it's struvite to us. It isn't a final proof, but together with the SEM analysis afterwards, as well as XRD, we can then conclude, yes, this is struvite. But this was non-destructive, and we could do this over time. So here's actually an image of one of these beautiful struvite crystals that was taken from the sponge specimen. And what you can see here is this is against the know-it-all database. So the black is the spectrum that was acquired, and then the red is the spectrum in the database. And you can see it's a really darn good match. So this really assured us, yes, what we see is struvite. This is looks fairly straightforward, is fairly straightforward, but still requires a little bit of pre-work, trying to understand how to do this inside the capillary. If you do it inside the capillary, this one was just a clean crystal on a wafer, basically. So we're also working on engineering methods, and instead of struvite, we precipitate out calcium carbonate. Calcium carbonate happens to have several morphotypes, aragonite, calcite, vaterite, and so on and so forth. But the same mechanism is basically used by us. We hydrolyze urea and then precipitate out a mineral. And we use this for various purposes, mostly environmental type of remediation purposes. And we've done this a number of times in the field now, that we drill a little hole into the well to go into a channel, seed the channel with our microbes or the enzyme, and then fill it up with biocement through this process where we repeatedly inject urea and calcium. So we're trying to understand how calcium carbonate grows in the pore space, basically. So we've done this on the microscale, and you've already seen these capillaries. This is kind of a porous media micromodel, all the way out to the field scale. And we've now been out 6 times and have done this successfully. It turns out that really these macro scale outcomes that we have, like plugging a leakage pathway, are based on microscale processes. So to understand the microscale processes is really important in order to develop better approaches on the macro scale, right? On a commercial scale, for instance. So I want to focus on the micro scale. And again, I've had the pleasure to work with lots of colleagues, students, and postdocs here who've done some really cool work and to have acquired some really beautiful images. So Nearger's rosettes, as I call them. There's some work by Logan that was published in Microscopy Today. 80s work where we assume that there are cells inside these little tubes, and some of Ellen's work where she has really beautifully demonstrated the association of these cells with these calcite aggregates. So again, going back to what would happen in a flow system where we want to plug something, we use these capillaries, and Subia, one of my PhD students, has worked with Fran Adar to basically, again, take these capillaries, and it's the same principle as I already explained in my previous slide with struvite. But what you see here is in the confocal, we can again image cells, red and green, as well as the crystal, but we don't know what it is. With the Raman scope, we can then go in and actually look at a crystal and get spectra. And again, there's glass, there's the liquid, and then we looked at two different crystals. One of them, or both of them, show us this 1086 peak, which is very characteristic for carbonates. Aragonite has a secondary peak that is about 701 per centimeter, the wave number. Calcite has a secondary peak that's at 711. So this indicates to us that this is most likely calcite and not aragonite that has precipitated here. Again, non-destructively, and we can do it on the microscale. For XRD and SEM and some of the other methods, we have to have a lot larger aggregate sizes in order to get that information. Now, again, this looks like it's easy. Well, it's not. Depending on how your crystal, your aggregate is oriented, you might or might not get a good Raman signal. So it is better to have a flat surface in order to do this, but it can be done even with naturally grown crystals, aggregates inside a flow system. Some of Neil's work produced these beautiful, as we call them, peach-shaped objects. And we thought it was calcite, and it might still turn out to be calcite. We don't know that yet. But so we've thrown a large number of analytical methods at this, and we're still not sure. But what Raman has shown us, we were able to zoom or focus on these peach-shaped crystals, as well as on this more amorphous material. And the amorphous material, we don't get a whole lot of the Raman spectrum. For these objects, we got something that is in the range of 1083, which is characteristic for carbonates, but there was kind of a double peak. So you can see them here. So what we did then is we took a optical calcite standard and acquired a spectrum. You can see it's kind of right between these two peaks, and a sample that we had identified as vaterite using XRD. And what you can see here is it really looks like that these materials are, at least on the surface that we can probe with this, mostly made of vaterite. We have not been able to find any vaterite spectra in a reference database. So we don't know that, but that's what I said earlier. Sometimes you compare it to standards that you have characterized to the best of your abilities. You can do this not only for inorganics, you can do this for organics. And I just want to point out, so Michi Wagner at the University of Vienna has really driven this forward quite a bit, the Raman spectroscopy in biology or microspectroscopy. And if you're interested in polyphosphate, polyhydroxyalkanoates, cytochromes, they all have a Raman spectrum. Now, if they're in a complex matrix, you might have challenges deconvoluting the spectra in order to figure out what it is. But what I'll show you later is if you can do that and you take these peaks that are potentially characteristics for these different compounds, you can then also create maps and say in a certain biofilm or in a certain tablet, as I'll show you, pill later on, you can show where the different compounds are. So you can do mapping. So now I'm moving on, and I will ask for help if I need to on some of the details. But Colin Shaw has done some work on the Raman scope, and basically what they are looking at is at this fault zone where there are fluid inclusions that can develop during the faulting, and they are using Raman microspectroscopy amongst other techniques to characterize it. So Colin showed me this beautiful image here of these potential fluid inclusions. And what they do is, among other techniques, they acquire Raman spectra and interpret them. So here are two spectra. It's kind of a very broad spectrum from about 600 wave numbers all the way to 3,800. And then there's a zoom in. So let's zoom into the top one. They see typical spectra, or that are indicative of nitrate, carbonate, as well as quartz, which all makes sense. But what they were really interested in was this area here where you have the hydroxyl group information that comes out. And if we zoom into this, so again, we zoom into this area, and I'm going to zoom in even a little more so you can see this better. Based on these OH stretches, the bands that are developing from that, they have concluded that there are different fluids that have been in different, should I say, times or different fluid inclusions in these samples that they had. And this is very low noise type of information. And again, it's just one way of looking at these fluid inclusions. This is not going to tell you that this was definitely a combination of sodium chloride and magnesium chloride, but it's an indicator that these are there, and it can guide additional analyses. Again, I always explain this to my students. If I hold up a bottle, you all see the Gatorade label. I don't know what it is. But if I walk around and I do this, so use a different technique, I get another viewpoint. And that's the same thing that we do with these techniques. SEM, Raman, XRD, they all give us a different viewpoint, different angle of our sample. And that's, take this as another tool in your toolbox, basically. So, Tayo, who is also working with Colin and the Energy Research Institute, they have looked at changes in mineralogy in samples from Kievan Dome, which is a dome that naturally contains CO2 up in north central Montana as part of the Big Sky Carbon Sequestration Partnership. And they have also used Raman spectroscopy, among other techniques, to look at rocks before and after exposure to supercritical CO2 brine mixtures. And what they basically saw, and these conclusions are, as far as I know, not based solely on Raman, is that there was no new minerals that developed during the supercritical CO2 exposure, but that there appears to be a recrystallization of certain minerals after or during CO2 exposure through dissolution and reprecipitation. So again, Raman is another way of looking at these samples and characterizing them. Connie Chang's group is currently developing basically a Raman-activated cell sorting in their microfluidic devices. What they mostly do, and Dimitri is doing most of this work, what they mostly do is they use fluorescent fluorescent detection to sort certain particles in their microfluidic devices. Basically, if they see a signal, they apply a little current and pull one drop here. If it doesn't have a signal, it goes there. Now, fluorescent markers are not always the most sensitive. They're not always the easiest to use. And Raman signals can be more specific. So they are working on basically developing a Raman-activated cell sorting. There is evidence that in surface-enhanced Raman, you can very sensitively detect some of these compounds that they want to detect. But they are really working on trying to do this in microfluidic devices. So what they are hoping to do is to use beacons, in this case, gold, basically nanoparticles that can enhance the Raman signal, and have those linked to certain pieces of nucleic acids that link up with certain viral populations, so that they can ultimately detect and sort them in their microfluidic devices. Our Raman Scope is not made to do that, but it is really helpful in developing the method, because what they do right now is they have these drop spots where they can actually collect these drops and then look at them. They're They're basically fixed in that drop spot, and they can develop the methods there. And their vision is to really have a whole spectrum of labels that they can use. With fluorescent labels, you have to have the right laser, the right filter set, and so on and so forth. So I am really curious how this is going to work out. But Dimitri has been pretty excited about his results so far. Nick Stady's group out of chemistry and biochemistry, they are working on phosphorus doped graphitic carbon. And they have worked with our scope quite a bit. And basically they're characterizing their phosphate doped graphitic carbon, again, through Raman spectra. What Nick tells me is that this peak out here is very indicative of graphite, and you can see the standard here. While these peaks are the D and G band, where the G band tells you something about the electric conductivity of the material. And again, these are important for fuel cells, electrochemistry, and so on and so forth. So what they did is they had different stoichiometries of carbon and phosphorus. And what you see here is, so PC3 and PC5 are different stoichiometries. And then you see the temperatures here. And what you see is that in this stoichiometry at 1050 treatment, 1050 degrees Celsius, you have more of a graphitic character, but you still have this D band that might be desirable or not. If you need more insight on this, I gotta refer you to Nick. That is not my area of expertise. And also they recently had a paper that is actually using that data. It's a little cut off, but the slides will be posted. And you can email me and I can send it to you. They also, then at 1050, where it seemed like they got more of this graphitic character, changed the stoichiometry between phosphorus and carbon quite a bit. And at some point, once they had enough phosphorus, they basically lost this graphitic character. And so this is not an identification of a material. This is a characterization of material properties using Raman spectroscopy. So it's not only can help you in identifying compounds, it can also help you understand how a material might change under certain conditions. Some people use Raman spectra changes to actually determine temperature, because there's very specific changes in the Raman spectrum for certain compounds at a certain temperature. The last example from MSU that I want to present here today is Cecily Ryan's group out of Mechanical and Industrial Engineering. And they're working on a very similar project, but what they are trying to do is they're trying to evaluate the use of biochar, which is basically a waste product of pyrolysis or hydrothermal liquefaction, and include that into plastics to make them electrically conductive. And that is being done with carbon black right now, which is really expensive, but if you could do it with biochar, it would be a huge cost saving. So what they're doing is they're using basically different weight percentage mixed into this blend of bioplastics. And then they are using Raman spectroscopy to again understand how electrically conductive this is. And this G band, again, is generally accepted around 1550 as an indicator of the electrical conductiveness of materials. So they acquire these and look at the intensities and how it changes with different percentages of these fillers, carbon black and biochar. So I was super, super excited when I saw a lot of this, when I got it, when I was to prepare for this seminar. I can't wait to hear more from others and see more people use it. It's available. And Erica, where are you? I know I saw you earlier. So Erica really deserves most of the credit on a lot of these data. She trained probably every single person so far who has worked on this. So let us know if you are interested. And I'm going to point out Dave too. He is always a good resource to point you in the right direction of how to approach your problem that you might have, your challenge. I want to close with a couple images that I got from Horiba. This is actually a pill that, and I forgot to look it up. I know it contains ibuprofen, but this is basically an elemental map. You use different peaks in the Raman spectra and you just map them across a couple millimeters here. And you can see that in these pills, there's filler, there's the medication, and then there's, I think there's a second active component in there. Here you have an image, and this looks beautifully resolved, right? Because it's a hard surface, it's plain, and you can look at it forever. This is skin, and skin is much more sensitive to laser exposure, and it's not quite as clearly defined, but you can still see the lipids, the pigments, and then two proteins in the upper layer of the epidermidis. So you can image this. You basically have a cross-section, see where these compounds are. And again, this is all based mostly on one or two indicator peaks in these spectra, these fairly complicated spectra that I showed you. We haven't done a whole lot of mapping at this point, but that's something we want to do. But with biofilms, while this is possible, you also got to worry about exposing your specimen for too long to a laser, because at some point it's going to burn. Not literally burn, but it's going to degrade. Again, this is one of those examples. Of course, this works great because it's a hard surface and very clean. It's a microfabricated silicon wafer. And again, here is an optical image, and then you can basically see how beautifully arranged this is with different materials here in this, on this wafer. And their big perk was we can do this in 2 minutes. So, and SEM and similar instruments, it takes a little longer. But again, it doesn't tell you everything either. So I want to point out one more thing, and this gives you a little bit of a background story on how we ended up with this particular type of instrument. I was going to get an entry-level type of confocal Raman. And when I was about to finalize my order, they said, oh, we have this beautiful high-end scope, and it's got a lot more than you asked for, and we can give it to you for just a little more. And we are very happy to have it, because instead of 2 lasers, I now have 3 lasers to deal with the fluorescence issue. We have better optics. There's a lot of advantages. But one of the items that I hadn't considered is the possibility to do polarized Raman. What that allows you to potentially do is actually to get towards the orientation of molecules and crystals, which is something that is out of my area of expertise, but Dave shakes his head. And I don't know how to do it. If you're interested in it, I'd love to learn from you and together with you how to do it. There's a few things that we got to do on the scope, nothing major. But if that's something that's of interest, this is possible. I had some slides about plastic analysis from Christine Foreman's group. I don't know where they went. It's some really cool work where they basically use snow particles, filter out microplastics, and then actually use Raman and other methods to identify that they are actually plastic particles. So they compare them to polypropylene and similar spectra. And it's been very successful. And I apologize, Becca and Christine, I don't know where they went. So, so back to this original slide. So we have different lasers. We excite a sample. We look at the signal. We split it up to look at the spectral information, and then we process it. Sounds pretty easy, is actually fairly easy, but just as so many other things, it is not going to tell you everything about your sample, most likely. It is one tool in your toolbox. For those interested in what this instrument really has, I figured I'd put this in the back rather than in the front and overwhelm you. It's an Olympus scope. We have 3 normal working distance objectives, 10, 50, and 100x, 2 long working distance objectives, especially if you have irregular samples. Please don't use my short working distance objectives because you can't scratch them. That's what these long working distance objectives are for. We have 3 different lasers. We have this fast scanning module, and again, this comes in for sensitive samples where instead of actually the laser moving or the stage moving, it's kind of an optical scanning where mirrors scan your laser almost like a sheet and acquire a certain plane at a very fast rate. Your signal goes down a little bit, but it saves your sample, especially if it's laser sensitive. CCD camera, and then this is actually pretty darn cool. So HORIBA promises to support users for the life of the instrument. That doesn't mean it's completely free. You might have to travel to their site, but they will arrange for a training with you. This is in our quote. Oh, yeah, the reason I have this is because I didn't talk about these parts in my results. This is in our quote, so we can hold them to it, but you might have to come up with the funds, or your PI might have to come up with the funds to travel there. I've had really, really good experience with HORIBA in discussing applications, and that's part of the reason why we went with HORIBA. Now, I called this earlier, I don't know if anybody noticed this, the new Raman. It's not the new Raman anymore. It's the old Raman now. As of about 3 weeks ago, we have a new Raman Scope, and this is more for biological systems. That doesn't mean it's not as powerful or less powerful on some of the things that I talked about. But Roland Hudson-Pichler, together with a number of us, was able to write an MRI grant about the time that I was purchasing the other one. And this one, I'm not going to take away all the details, but this one has laser tweezers, basically, where we can pick out single cells. And Roland is going to give a TBI seminar on March 11th to talk about the details. There's also some potential seed funding for biological work available. So if you are curious about Raman, Roland's presentation is going to touch on more the biological parts of Raman microspectroscopy. Please join us for that seminar as well. Here, I'm just going to leave this up for a moment. These are links. I can send them out. I don't know how we can do this through MONT, but I'm sure we can post them on the website with videos and information that you might be interested in on Raman Spectromicroscopy. And I want to close with saying thanks again to those people and entities at MSU who made this possible. Without you, I couldn't have purchased this. So thank you, thank you, thank you. And with that, I'll take any questions. Kristen.
What do you think the limitation will be in terms of depth?
So the question was, what limitations will there be with respect to depth, especially for people who do biofilm work? Again, that depends on the absorptivity, if you want to so, of the material. You can hit them with a lot of light and you can get deeper, but the Raman signal is often less strong than the fluorescent signal. And we have observed depth limitations with the fluorescent signal. So you're pretty much facing very similar limitations. What Raman spectroscopy can do for you is that you don't have to stain your biofilms. You get chemical information from it, and you don't need to pre-treat it. And it's, again, just like so many other things, it gives you another viewpoint of your sample. And what we're hoping to do at some point is to be able to do correlative microscopy, where we can take the same sample, put it on another stage or put the stage on another scope and image exactly the same spot. And that's kind of, it's really not that far out, hopefully. Does that roughly answer your question?
I don't know if you want to tell us out any numbers.
No, I can't, because it depends on the density, right? Yeah, or yeah.
So, do you have any minimum sample size requirement to get a good Raman signal, or is it just like size of the beam, laser beam that you have, small as that?
Yeah. So the question was, is there a minimum sample size that you need in order to get a Raman signal? So again, this is theoretical, plane surface, good signal, not a whole lot of scattering into other directions. It's claimed that you can go down to a micrometer or maybe even less. I know some of our peach-shaped objects are in the 3 micrometer range, and we've gotten beautiful spectra off of it. And they are not plain, I can tell you that. So it's cell level, but maybe not subcellular level for prokaryotes. Yeah.
Very nice. I'm wondering if we can analyze the quantitative, not qualitative analysis using this. And another question, how it could be different with FTIR or APR?
Yeah. So the question was whether there's a possibility to analyze things quantitatively and how this compares to, or maybe is complementary to FTIR and even ATR FTIR. So I would be very careful in extracting quantitative information from these spectra. That doesn't mean that it's not possible if you do it right, but at this point, it's qualitative. At least for the work that I have presented here and what we have done. It is very much complementary to FTIR, and as a matter of fact, we're waiting to have not in my lab, but to have an FTIR microscope and an FTIR, ATR, FTIR installed. So there are 1 or 2 on campus that are a little older, but this should help us getting, again, more insight. And again, just like Raman, FTIR is non-destructive, and you can take, get a Raman spectrum, get an FTIR spectrum, and then go on to XRD, XPS, Auger spectroscopy, whatever you decide to do. So these are great techniques to get started on a sample because you're not destroying your sample unless you shine a laser on it for too long. And you do see decolorization when you have your laser on for too long on the samples, on some samples anyways. Any other questions? If not, I know Heather has distributed questionnaires. Please fill those out. If I didn't say that, she'd let me know afterwards. And if there's any feedback that you have beyond what you can put on those questionnaires, please let David, Heather, or me know. Thanks, everyone.